inviting me. It's always really nice to come to ICRISAT, especially when it's nice and sunny and warm. What I'm going to do is I'm going to really change the sort of scale and pace a bit here, because although we have lots of interest and we're very interested in the areas that have been covered so far, one of the things that's become an increasing issue from our point of view is really underlying quality of the data, and this is something which we find can be a little bit challenging, but it's also nice to think a little bit about the underlying quality of the data. I really just want to use this talk to highlight some of the issues that really focus around when is a SNP a SNP. So, you know, as we've been hearing up to now, the new sequencing technologies are providing great opportunities, and not only in crops that have a large resource base, but can be really quite widely applied in a whole range of crops. You can generate data very high throughput. They're cheap per sequence or genotype, and they're becoming increasingly cheap per sample. And it's the per sample cost, I think, as people have highlighted, that's often the main driver in terms of breeding applications. But it's one thing that's really important, and this is something which affects us greatly because we work with a wide range of different species, is the opportunities and cost effectiveness vary by organism. And some of the key elements that affect the application for these things are things like genome size. If you're working with a very large genome, there are some problems in terms of what you can do in terms of sequencing. The number of reads you have aren't so efficient with very large genomes. Polyploidy is clearly a complication. Heterozygosity is a complication. And really, so in order to get around this, again, there are a number of genome reduction methods that people are using. Some we've heard about already. And simply because in many cases with large genomes, resequencing, as in total genome resequencing, is often not cost effective. And again, there are a number of methods around. Originally, when we were just wanting to look at a few samples, doing RNA-seq, just sequencing the transcriptome, is one method of genome reduction. You've heard about GBS or alternatively RAD sequencing, where you're sequencing off of restriction sites. Or again, you can use exome capture. And these things are often combined with imputation strategies. And this is really all about the cost-effective use of the technology and the species that you're working with. So one of the things that really started to interest us very early on was the complications really arise around when is a SNP a SNP, the issue about false positives in terms of SNPs in your analysis. And this can have potential implications for downstream analysis, and I think one of the challenges is really to work out to some extent what the complications are for downstream analysis. But from our point of view also it has financial implications. So for example, if you go through sort of SNP discovery for developing genotype platforms, if a lot of your SNPs are false positives, you're wasting samples or slots on your genotyping array, and that can become a big issue. And we had a couple of scares early on when we were doing sequencing with next generation sequencing technologies, and we generated some genotyping arrays, and they turned out not to be very good. And we got a lot of flack for doing that. So one of the things that we started to think about is, can we think a little bit about the quality of the SNPs? And when we started doing this, I mean, a number of my colleagues on the molecular biology area said, well, it's fine, you just run this stuff through a pipeline, you find out what's a SNP and what's not a SNP, and it's not quite as simple as that. And I really want to focus on some of the issues and with a little bit of illustrations. So one of the causes you get in terms of false positive SNPs is mismapping of reads. So a lot of the discovery or genotyping is done by mapping reads onto some form of sequence. And one of the questions it really comes down to is your read mapping in the place it really should map in terms of the genome. And there's a number of reasons why that might not be the case. For example, if there's recently duplicated genes, and there's certainly many cases in particular in plants where you can get recent tandem duplications of genes, and that can cause a problem in terms of where read maps. And this is some of the symbols you can see is that quite often you get heterozygosity where you didn't really expect heterozygosity. You often find SNPs in very strong linkage disequilibrium. This is just an example. And quite often you get two or more clearly distinguishable classes of reads with variants which are in phase. in the case. And one potential way of doing this or coping with this is really the mapping tool you use can be a big factor. And what's happened over the years is the quality and performance of different mapping tools has improved as people have thought a little bit about it. And both the efficiency, computational efficiency, and the quality of the mapping has changed. And again, there's a number of studies where people look systematically at different mapping tools and their efficiency in working. But it's also important to realize that you can actually mismap reads quite legally as well. So really one of the problems you have is that, you know, if you have a situation here, I've got a sort of a naughty situation here where you start off with a couple of reference sequences which differ by a SNP. And if both the references are there, when it comes to mapping these trivial short reads, then everything maps really quite nicely. But one of the problems is that if you have a situation where you don't have a reference, you've got essentially one of the sequences missing from your reference because you've only got a partial genomic sequence, then you have a problem then, because if you're allowing for one mismatch in order to detect SNPs, then you get a situation where essentially two different sorts of reads map to the same position in the genome. So you've got to think a little bit about this. And so this is the sort of situation that you get. So if you have an ideal genome sequence here, you've maybe carried out a whole genome shotgun assembly, and this is the case with many crop species, as you've heard, where you have at least regions of the genome which are assembling really quite nicely, but you have perhaps some regions that are missing, and these are candidates for if we don't have as it were, the home for these reads, what happens to them? In some cases, they'll disappear completely, but if they've got a certain degree of sequence similarity, they might mismap into the genome. So one other issue, and this is that there are actually sequence-specific sequencing errors. So Illumina sequencing, for example, is not perfect. There are particular sequencing motifs and types which Illumina doesn't really cope with that well. And one of the problems is with systematic sequencing errors, and as opposed to random sequencing errors, is they accumulate. So essentially you get multiple errors that all tell the same story. So that can cause a problem for you. And one of the nice things, though, if you're wanting to try and identify these, is that you can, they tend to all be in the same direction. So reads in the same direction have a problem, reads in the other direction don't have a problem. And this is just a view where we've actually colored the reads by the direction of the, in which the read actually takes place. And you can see that in this particular case here, we've got multiple cases. These are actually false positive SNPs. And they're all occurring in one read direction. And they're not occurring in the other read direction. The other problem, and again this is an issue, is that you can get issues with misassembly of the genome. So again, if in particular where you've got relatively sort of quick and easy or quick and dirty genome assemblies that you're working with, and that's often the case, and I really want to emphasize that many people are working with such genome assemblies, then you can get a problem, that you can get a situation where there's actually an error in the reference sequence. And there's a number of problems that you can get with that. So this is an example here where we've essentially got a region in the genome which has two different regions which are relatively similar. The assembly hasn't worked out cleanly, and you've essentially got two different regions have collapsed together in the genome assembly. What happens is then you've got a reference which you've got a faulty hybrid reference sequencing which has been generated by optimizing the sequence from both read types. In this case, with strict mapping, what you find here is that all of the reads in this case show a difference from what is the false case. Again, with more relaxed mapping, yes, it sort of works, but you get essentially lots of mismatch reads occurring here. So there's quite a number of occasions just from the mapping side of the process where you can have problems. One other issue that occurs in sequencing, it's less of a problem now than it used to be, is you can generate optical duplicate reads as part of the sequencing process. And again, when that's the case, you get multiple identical reads coming from the image analysis. Again, in the case of where you have multiple sequences spread across a region, you can tend to identify these really quite nicely as they come out as being identical reads, and there are tools that you can get rid of duplicate reads for. But in terms of some genotyping technologies, there are some problems that occur with this. So if you have something like GBS, where you essentially have all your reads in one direction, that can be a problem. So I want to really just show a number of examples that just sort of highlight some of the issues around this. This is we ran a little test experimental system where we took some data from an inbred. In fact, this was some public data from a maize inbred. We took RNA-seq data, and what we did from this RNA-seq data set is that we generated our own reference assembly from this. And actually, I will say that depending on which which assembly method, you can find differences depending on assembly methods. We then mapped the reads back onto the assembly and then did SNP discovery. At the time, we tend to use Freebase as our SNP discovery tool. And then one of the things that we do a lot of, and my group's quite well known for visual inspection. We like visualization because we like to see what we're actually doing. We like to understand what's going on under the hood. We looked at, in this sort of process, we looked at a number of things. We looked at the read length. In this case, from this data set, we played around with the read lengths, either 50 base pairs or 80 base pairs. We've now done a lot of simulation now to look up to a KB. We used trimmed or untrimmed reads because, again, that's one of the issues you might have to deal with. We looked at mapping modes, and a lot of read mappers have a number of different mapping modes. One of the questions here that you have to deal with is that if you have a read that can potentially map to more than one place in the genome, what do we actually do with this? And so it's an interesting question. So there are some mapping modes that allow you to say, well, if it maps to more than one place in the genome, we'll just map it every place it maps. In some cases, they'll map it to the first place it finds that there's a match. In some cases, you can throw away all the reads that map to multiple places in the genome. So there's a number of different mapping strategies, and some of those are available in some read mapping tools, and some are available in other ones. We played around with a number of mismatches, and that can be really important as well. In this case, we used 1 or 3 mismatches. And we looked at the difference between using paired end reads and single end mapping. And we produced all different combinations of these. Say we use Freebase for this, but we have a parallel version of Freebase that we've doctored in-house, which improves the efficiency for these simulation processes. And really the take-home message is, well, one of the things we had to do, obviously we had to do some normalization, because making these different approaches comparable takes a little time to think about. And the key take-home from this is virtually everything you do in these cases can affect the number of SNPs you can get. And you can get lots of false positives from all of these causes. And so it's important to realize that that's the case. So one of the things that we find, again, because you're using an inbred, and one of the nice things about using an inbred is that you can find SNPs, if they're false positives, quite a lot of them are heterozygous. So you can say there's something a bit strange here. I know this is an inbred line. So if you've got SNPs that are heterozygous, then there's something of a problem. There's SNPs that come from misassembly, and there's SNPs that come from sequence-specific sequencing. So basically, that's really sort of quite an issue. We have played around with the mismatch parameters. We played around with paired end as opposed to non-paired end, and we played around with trimming. So really, the take-home message is that really pretty much everything can generate some degree of false positives, and so it's important to do that. Okay, I'm going to skip with this. So one of the things I wanted to really talk about again is a bit about mapping mode, and this can be really quite important. Is that and one of the things is that some of the intelligent SNP callers really use information about the quality of the SNP calling. So in some cases they'll just look at the straight reads, but in other cases they'll be able to utilize the quality of the mapping as part of the calling, and that's really important. So there are some naive SNP callers around that many people use in their pipelines, and that can be something an issue. And actually, some people use just visual. Is it a SNP? I can see what looks like a polymorphism. So again, just to illustrate some of these things on some real data sets, Neil Stein talked a little bit earlier about the Barley POPSEQ data set. So this is a resequencing data set from Barley. It's based on resequencing a mapping population. And so one of the things we've had a look at this data set from is we've done some visualization with it. We've loaded all the called SNPs from this into our graphical genotyping application called Flapjack. And when it does this, so these are a number of different lines down here. There's I think 90 odd lines here, and these are the different SNPs along the top here. In this case, they're coloured by the nucleotide of the SNP call. What we can do with this is we can select a particular sequence contig from the MOREX assembly. So here we're selecting the SNPs from a particular contig. We can then colour up those SNPs relative to the reference genotype, which is MOREX. So everything here that's in this set here is coloured relative to MOREX. And what we can do in this application is we can then sort the genotypes. So this is a relatively low resolution panel, and so what happens when you sort relative to the genotypes, virtually everything in an individual contig looks identical, i.e. it's either MOREX or it's the other parent. But occasionally you get these things here, which where there's a problem. It looks like there's some of the there's one individual SNP in the contig that's telling a different story. Just to show the scan along, this is a larger example. This is looking along the larger data set. If you expand up on these things, what you see is that these problem SNPs here are flipped round. They've been flipped round the other way, and these are SNPs where there's essentially a genotype, or there's an error in the reference assembly. So the MOREX reference here is wrong. So when you map back onto it, you're ending up with your SNP call is the wrong way around, and you can see that really nice and cleanly. But one of the issues that you face is that in terms of mapping is that what you do with really high density data sets. And so this is a data set. This is a data set of Solanum. So this is a data set with a number of different species related to potato. And what we've done here is this is an RNA-seq data set, and this data set is based on SNPs from I think there's 8 different species that we've actually taken RNA-seq from 8 different species. And then the big question is, first of all, what do you map it back onto? So we have a reference sequence for potato, but it's a diploid homozygous potato, which is really nice for sequencing. But one of the issues then is what do you map it back onto? And when we map these data back onto the reference sequence, you can find lots of polymorphic SNPs. But one of the issues is that what mismatch threshold do you use for this? Even in cultivated potatoes, there's probably a SNP every 20 nucleotides in genes. So the mismatch calling rate that you use is really quite a big issue in terms of whether you're actually mapping accurately to where the read should be in the genome, or whether it's someplace in the genome. And really think about this is that if you're working with read lengths that say around about 100 nucleotides or less, then one mismatch is 1% difference is an issue. Then if you've got maybe a SNP every 20, then you're talking about maybe 5 mismatches in order to map something back even to where it should be in the genome. So there's really an issue around that. So again, this is another example, just again to show some visual data. This is some data from blackcurrant. So blackcurrant, again, when people were talking about relatively minor crops, blackcurrant is a relatively minor crop. There's the number of people working on blackcurrant genomics around the world, I think, is 2. I think at the last count. And you can do a lot without a reference genome. So this is some data we were working with in a genotyping by sequencing study. This is using a mapping population, and these 2 colored blocks here are the 2 genotypes that are the parents of the mapping population. And again, this is a case where we're working with a heterozygous material. So we've got a cross between 2 heterozygous genotypes. And so if you've got a reasonable read depth, and again in the parent you can see this, this looks convincingly like a heterozygote here. at this position here, this looks like convincingly like a heterozygote there, this looks convincingly like a heterozygote there. But then you start to wonder here, well, I've got two potential variants there. Is this line here, even with that read depth, is it heterozygote or not? And you can give a sort of quality decision based on some acceptance of threshold. But then if you actually start to look at the population, and if you want to look at a large number of individuals, then you start to think about, again, I've coloured up individuals here, it doesn't show up tremendously well, but different individuals here have a different block of colour. So again, you can start to say, well, this individual here, yeah, I could be convinced it might be heterozygous here. This one here, well, I've got one variant. Is this really a heterozygote, or is it just a chance variant? But one of the nice things, again, working within a population where you know what went into the parents, you can use an inheritance test very nicely. So that's a really powerful tool when you're using this sort of technology, where you can constrain what's gone in, so you know that what you should have in your material is constrained. Clearly, it's a much bigger issue if you're starting to use this type of technology for looking at large-scale germplasm search surveys. Okay, so I'm going to try and summarize here at this point and give a chance for some questions. So there are clearly a number of factors that can contribute towards false positives, and I don't want to depress people. If you think quite hard, there's things you can do about it. You can look at the read depth in which you're sequencing. You can look at the read length, which can help in terms of mismapping. The quality of the reference sequence is an issue, but that's changing all the time, even in many sort of low resource crops. You can use different mapping tools, and you can work with different mapping parameters and analysis pipelines. You've got to watch out, or at least be careful of technology-specific errors, and you can build that into your pipelines. You need to think a lot about the biology of the target species you're working with. So, the worst-case scenarios are things which are highly heterozygous, things which are very large genome size, and things which are polyploid. Unfortunately, you can get things which are all combinations of these, which can be something of an issue. What you can do, though, is you can use various forms of aggressive filtering of your data. So you can identify SNPs in the first place from various methodologies. But what you can then do is you can say, well, I think there's a SNP there. The data seems to suggest there's a SNP there. Is there any reason that it might not be a SNP? So again, if you can use inheritance tests, you can use heterozygosity tests, you can use sequencing motif tests to look for this. What increasingly, if you're starting to accumulate a database of information for your species, you can also look and say, well, is this SNP being discovered in this species already? Is there evidence already that this position is polymorphic? And there's actually some interesting publication recently on sorghum, which is starting to accumulate that sort of data. So one thing, and really it's also important, is to validate these analysis pipelines for each application. So there are some cases where false positives are not so much of an issue. In that case, it doesn't matter too much. But there are some other cases where you really, the fact that you may have called a SNP wrongly is a problem. There's also potentially problems in the reverse of this. So one of the issues is that if you use aggressive filtering to be able to identify SNPs, the other side of it is by doing that, you also will miss a lot of SNPs. So there'll be a lot of false negatives in your data. So you'll get very robust SNPs, but you'll also miss a lot of SNPs that are there. So you need to think about the application and the biology. And so that there may be consequences depending on the downstream analysis. So really, the take-home message from all of this is that false positives are really there. It's really important, I think, not to use analysis pipelines without thinking really quite hard about, is this pipeline optimized for the species and the situation that I'm working with? What are the consequences for the particular biological application that I'm working with of having a false positive? But also realizing as the technology is improving, this is very much a moving target. And certainly in our experience, the analysis pipelines we're using now seem to change at least every few months as we accumulate better reference information, but also as the technology improves as well. Okay, and just to say that there's a whole range of people. I've just highlighted some of the people really immediately the James Hutton have been involved in some of this work, but I want to emphasize that this is all the work that we do is dependent on links with a number of major programs around the world. Thank you very much.
Thank you, Dave, for that very informative presentation, and particularly your summary was quite revealing. We have time for a few questions.
Silence room. Yeah. I mean, the issue is that if you have a read of something like a read length of 100, you know, that will not always go to a unique place in the genome, and so the probability of that really depends an awful lot on the nature of the sequence and the nature of the sequence context in which it's in. There's obviously clearly cases where you've got repetitive sequence that can be a major issue for that. But there are many cases where you get recent tandem duplications of gene families are really quite common in plants, and that can cause an issue for that.
Thanks for the valuable suggestion how to avoid, positive or SNP phenotyping. What about haplotyping? Use of haplotype and SNPs to avoid all these troubles and still be very comfortable whatever the approaches you have for SNP genotyping. Use of haplotypes. They will really reduce all these problems whatsoever I have highlighted.
Again, I think haplotypes can help in that. I mean, one way you can QC this is by essentially doing imputation and really checking with imputating. You can do a leave one out and then impute. So essentially, the haplotype context can be really quite valuable as far as that's concerned. But again, how robust haplotyping is will depend an awful lot on the extent of LD and the background information you have on the species you're working with.
How strongly you believe in imputation of GPS data? Have you ever validated the imputed GPS data with the resequencing?
Well, I think resequencing and GBS both can be affected by quality. So one of the issues you really face here is what is the baseline you're working against.
Any questions from this side?
Yeah.
Okay. Both.
So your pipeline and all these based on Illumina sequencing platforms. So what about other platforms like Ion Proton or your base calling?
Yeah.
So what you said totally mostly on Illumina, if I understand. What about other technology platforms? Which one is the best?
I mean, I'm very happy with Illumina sequencing. I don't have a problem with it, but I also like to know or I like to be able to play around with exploring the quality of the SNP that I'm dealing with. I mean, one of the things that we've done again, just to validate as well, is cross-checking against genotyping platforms as well. So that can give you a gross check as to whether you have a problem. And again, if we aggressively filter, we can get a really, a very good and very tight association between what we can detect by NGS-based sequencing and chip-based genotyping. But again, in doing that, we're identifying a subset of really robust SNPs. And there are clearly SNPs as a result of that you're going to miss. So again, cases where you have multiple SNPs close together are going to fall out by that sort of filtering. Also, also what can affect this as well is what reference you're mapping onto as well. So again, one of the things that we're starting to look at is playing around with a whole range of different references. And in some cases, it may be actually better reassembling rather than mapping as well. So where you can essentially reassemble within a genotype, provided you have enough depth of sequence to be able to do that. Okay.
I don't see any more hands over here, and let's thank